Let's now focus on some distinctive characteristic of picornaviruses as well as pathogenesis of some of infections they cause. So the most important and impressive changes the viruses, picornaviruses cause in host cell are inhibition of the host cell macromolecules, and induction of cytoplasmic vesicles. So what is outcome of the inhibition of host cell mRNA translation? So cell cannot synthesize proteins, macromolecules, because the virus is shutting down the cap dependent translation. And we know that cell cannot survive without translation, without synthesis of protein. How does induction of cytoplasmic vesicles, you recall those vesicles, serve as a docking site for the machinery which is involved in the viral replication? Well, because they reform the vesicle inside of cells, they alter normal intracellular transport pathway, which also contributes to cell death. Now, poliovirus, we said that approximately 1% of people who are infected with polio develop paralysis. In 90% of infection, there are hardly any sign. In approximately 8%, the infection is manifested by fever, headache, those general symptoms, flu-like symptoms. However, that paralysis is serious. So virus enters via mucosal surfaces. We know that it follows the orophycal route of transmission. In the mucosal, virus replicates and gets into lymph nodes, and from lymph nodes it can be disseminated as viremia. And basically, if that extra neuronal tissues virus replicate, but as a neuronal motor neuron can be infected in the tissue. And via retrograde transport, you recall the microtubule pathways, dynein. So via this retrograde transport, the virus can enter spinal cord. And since it triggered death of cells, that's how it causes paralysis. And in 1950s, there was huge endemic of polio infection in the United States. More than 50,000 people become ill, and 20,000 of those were paralyzed. That's one those which were paralysis that were recorded. And this paralysis didn't affect it only motor neuron. It affected also smooth muscle. So not just voluntary movement, but involuntary, which caused paralysis of muscle required for breathing, diaphragm. So these people could not breathe on their own, so they were placed on this iron lung. I'm sure you never even saw any of those. So those iron lungs helped people to breathe. If you are interested in this, there is a reading posted on Canvas, which is um, kind of the testimony of patient who got infected with polio as a young boy, and all his life he suffered from this problem with breathing and had to use iron lungs. So, since this was such a serious outcome, even so, it affected only around 1% of infected people, but the outcome was really serious. A lot of effort was devoted to develop vaccines. You remember the March of Dimes organization, research, and so on, focus on developing a vaccine. And two vaccines have been developed. One so-called Salk vaccine, which is inactivated. So the virus was prepared and inactivated by formalin. And Sabin vaccine, oral vaccine, which was attenuated vaccine. So Sabin vaccine was much more efficient. However, there was a big problem because there was only a single nucleotide mutation in RS stem loop 5. And if you have single mutation, what might happen? it can reverse. And it was noted already that outbreak of poliomyelitis in Hispaniola associated with circulating type 1 vaccine-derived poliovirus. This was one of the first paper which pointed on the vaccine-derived poliovirus infection. And why does this happen? You recall this loop 5. So there were three different Sabin vaccine, Sabin 1, Sabin 2, and Sabin 3. Each one had different mutations. They were attenuated through passage to semi supportive tissues, but each of them had only one nucleotide mutation. And since it was in this iris, basically the virus couldn't properly dock on ribosomes and could not replicate. However, this one single nucleotide mutation could be reversed. And this reversal created fully infectious virus and caused what we call VAPP, vaccine-associated paralytic poliomyelitis. And because of this, basically, this Sabin vaccine was discontinued in USA in 2000. So don't be scared to get vaccinated because the Sabin vaccine was discontinued in US. And this is basically a report from CDC, which it shows all these cases are vaccine preventable disease. This is vaccine relevant poliomyelitis. So we don't use Sabin vaccine anymore. In US, we use Salk, which is inactivated vaccine. Now, another important virus which belongs to picornaviruses is hepatitis A virus, or infectious hepatitis is caused by this virus. It's distributed worldwide, and as I mentioned before, it is transmitted via contaminated food or contaminated water. That's why whenever you go to travel to area of the world when hygiene is not sufficient, always be careful what you eat, what you drink, and the best way is to get vaccinated before travel. Um, HIV, hepatitis A virus shares only 28% identity with other picornaviruses, but his genome is very similar. It has iris, and it also triggers this CAP-dependent inhibition of CAP-dependent translation. Um, Infection with hepatitis A usually is self-limited. It means it's clear by itself. However, approximately 2% of adults, older adults, can develop some serious condition where the liver can be destroyed, and it can cause severe liver tissue damage. Now, as I mentioned before, maybe hepatitis A is infectious virus, which is part of picornaviruses. Hepatitis C is flavivirus, and hepatitis B is hepatnaviruses. So even so, all three have so-called similar name, hepatitis virus, they all belong to different families and have different pathogenesis. Now, here is one study. There's a study efficacy and efficiency of hepatitis A vaccine. This was double-blind, randomized study in Thailand, and it involved over 40,000 children which were vaccinated. And basically here you can see time after vaccine on X axis, and y-axis shows the cases of hepatitis per 100,000 population. So basically, this two arrows shows the vaccination, and this is time after first dose. So they started to over 100 days later, they started surveillance. And here, this arrow shows the booster vaccine. And you can see on the bottom number of cases in that in the population that was vaccinated, and this upper curve shows number of cases in those who receive control or placebo vaccine. So this clearly shows efficacy of the hepatitis A vaccine. And this control vaccine here, it was actually not placebo, it was hepatitis B virus, okay, vaccine. So vaccination is really efficient, and whoever travels to part of worlds where the hygiene is low, it's strongly advised to get vaccinated before. Now, let's talk at the end about rhinoviruses. We know that the rhinoviruses cause more than half of common colds. Other half is caused by coronaviruses, enteroviruses. Human rhinoviruses, actually, I mentioned before, there are over 100 different serotypes, and that's why we don't have vaccine. And they replicate only in upper respiratory tract. It's very rare, almost undetected, to get new infection of the lungs or lower respiratory tract as rhinoviruses. And it is because rhinoviruses will replicate well in lower temperature, 33 degrees. And basically higher temperature of our body, 37 degrees, um, do not support replication of rhinoviruses. So it's very unpleasant, but it is not usually dangerous until it becomes. And there is another paper which I posted, which shows that in some cases, infection by rhinoviruses as children might cause serious problem.